Hey guys, I'm back to talk a little bit about the structure of DNA, and I hope you recognize these people over here on the left-hand side, Watson and Crick and Rosalind Franklin, who are credited with figuring out what the structure of DNA actually looks like. Now, when you talk about the structure of DNA, it has the classical double helix shape. Double helix means it is a ladder that is twisted. Now, why it gets the name for ladder is because it has a phosphate and sugar backbone, okay? And these rungs are made by nitrogen bases, and then it's twisted like over here. It's twisted, so that's where it gets its double helix shape. Take the ladder and twist it is what makes the double helix shape. Now, when we talk about DNA, we've got to understand the directionality of DNA. DNA has a 5' end and a 3' end, and it's all linked to the carbons. DNA, or the ribose sugar here, has 5 carbons, 1, 2, 3, 4, 5. The 5th carbon is kind of hanging off. That's where the phosphate's attached to. All right. The 3rd carbon is down here. That's where the OH group is attached. Now, the thing that you need to know about that is that whenever a nucleotide attaches, so this is a nucleotide here, right? Whenever that nucleotide attaches to the nucleotide down here at the bottom, they always attach right here. The 5' end from the nucleotide being attached goes here. So let me kind of understand which way. The DNA that is in existence right now is up here. This one is the new nucleotide that is being added. And if you'll notice, the 5' is attaching to the 3'. So really, DNA grows on the 3' end. It's going to keep adding. The 5' is going to come up and add. They're going to keep going. So this DNA in question is growing in this direction. It's getting longer and longer as you go down. That's going to be important later on. So as you can see, when you look at a DNA molecule, it actually has anti-strands. You have a 5' and 3' end this way, and on this side you have a 3' and a 5'. They're opposites of each other, right? So this one is here, this one is here. Now they're complementary strands. They're running opposite directions, basically. Now when you look at the DNA and how it's bonded, there's a couple of bonds that you need to know about. Hydrogen bonds are what are formed between the nitrogen bases. Whoop. Sorry about that. Formed between the nitrogen bases. And think about these bonds are really weak. Now, the covalent phosphodiester bonds are what is formed between the OH group and the phosphate. All right? And those are pretty strong. So if you think about it, we've got strong bonds along the backbone, and we've got weak bonds along the rungs, which really promotes the idea, which allows for it to split and be replicated pretty easily. Now, we also need to keep in mind that there's purines and pyrimidines found within DNA. You've already learned about those, but just a refresher. All right, so your purines are A and G, if you remember, like pure gold, right? Purines. And your pyrimidines are T and C. Remember, a purine always goes with a pyrimidine. Purine always goes with a pyrimidine. Now, A's and T's always bond together with 2 bonds, and C and G's always bond together with 3 bonds. Now, the last question we have to ask ourselves, which is what the whole video next time is going to be talking about, is how is DNA copied? How does replication of DNA happen? Well, because of the way it is designed, once you break that bond, those hydrogen bonds down the middle, you end up with 2 strands, right, that are opposites of one another, but they both allow for nitrogen bases to come and bind to them in order to create 2 new strands. Now, the good thing you need to look at with the 2 new strands, that each one of these has an old strand, and each one of them has a new strand in the new DNA that has been made. Very important. And that is why we call it semi-conservative copying. It's because when it copies, half of the new is there and half of the old, so it conserves half of the old. Now, you might see sometimes a thing like the conservative model and the dispersive model. The conservative model is like over here, which basically said if it worked this way, you would keep the old strand of DNA and you would create new strands. We know this is not what happens. The dispersive way is where the new ones would have parts of new and old, and that doesn't happen. What really happens is right here. The semi-conservative model is correct, and it was done through experiments by Milsison and Stahl, and it supported what Watson and Crick had said earlier. All right, in our next video, we're going to talk a little bit about replication of DNA, and I look forward to talking to you then. Have a good day.